And the purpose of that pathway is to oxidize acetyl coenzyme A to carbon dioxide and to recover the energy from those oxidation reactions in these reduced cofactors, NADH and FADH2. So from each oxidation of an acetyl-CoA to 2 carbon dioxides, we reduce 3 molecules of NAD to 3 NADHs. We reduce 1 FAD to FADH2 and make 1 GTP from GDP. We can think of that as an ATP equivalent. So we're going to talk today about what happens to those electrons transferred from acetyl-CoA to the reduced cofactors and how they're used to start to produce ATP. We started to talk about on Friday that the electrons are transferred to this molecule, coenzyme Q, which is also called ubiquinone. So it's a quinone, a ring molecule with a quinone. This is this carbonyl in pair positions from each other. And that accepts electrons from FADH2 to make ubiquinol. So these double bond oxygens are then reduced to hydroxyl groups to make ubiquinol. And then these are carried forward to the electron transport chain that we're going to talk more about today. As NADH and ubiquinol are oxidized, that happens in mitochondria, and we're going to talk about that. It happens through protein complexes that are in the inner mitochondrial membrane. That provides energy for synthesis of ATP. So oxidation of NADH and QH2 provides energy. The oxidation of 1 NADH provides about enough energy for synthesis of 2.5 molecules of ATP. And the oxidation of 1 ubiquinol, the ubiquinone, generates enough energy for making about 1.5 ATP molecules. So we can start to do some calculations of how many molecules of ATP can be synthesized using the energy from oxidation of acetyl-CoA. So if we start with 1 acetyl-CoA, go through the citric acid cycle, the isocitrate dehydrogenase and the alpha-ketoglutarate dehydrogenase, the 2 oxidative decarboxylation reactions in the citric acid cycle, each generate 1 NADH. So each of those then can be used, that energy is used to make 2.5 ATPs. The succinyl-CoA synthetase step makes a GTP molecule directly, which can be converted in cells to an ATP. So 1 ATP from that step. The succinate dehydrogenase step transfers its electrons to QH2, so we get 1.5 ATPs there. And the malate dehydrogenase step generates directly an NADH, and so that uses and can be used to generate energy for another 2.5 ATP. So from 1 molecule of acetyl coenzyme A, we can gain enough energy to synthesize about 10 molecules of ATP. So we're really getting the energy out of that acetyl-CoA acetyl group by oxidizing it completely to carbon dioxide. So let's think about it if we start from glucose instead of from acetyl coenzyme A. So we're starting with glycolysis. We know that there's a net yield of 2 molecules of ATP in glycolysis. And there's 2 NADHs that are produced. And if we're using the citric acid cycle, then those NADHs can generate energy for making 5 ATP. So from glycolysis, we can gain enough energy for making 7 ATPs. The pyruvate dehydrogenase complex makes 2 NADHs. So remember that decarboxylates pyruvate to make acetyl-CoA, and there's 2 of those from each glucose molecule. So we get 2 NADHs, and so that's another 5 ATPs. And we just said that in the citric acid cycle, from each acetyl-CoA, we get enough energy to make 10 ATPs. Because there's 2 acetyl-CoAs from each glucose, then that comes out to 20. So from complete oxidation of one glucose molecule to carbon dioxide, that generates enough energy to make 32 ATPs. So it's an efficient pathway for extracting the energy from that sugar and using it as an energy source for making ATP that the cell needs. Okay, there's a nutritional disease called beriberi that people get if they don't eat enough vitamin B1. And another name for vitamin B1 is thiamine. People who are not getting vitamin B1 from eating fruits and vegetables can get neurologic and heart symptoms. And one of the things we can test for that is that there's an increased level of pyruvate and alpha-ketoglutarate in the blood of people who have beriberi. So I want you to think about how does a deficiency in vitamin B1 result in increased levels of pyruvate and alpha-ketoglutarate in the blood. So talk to each other about that for a few minutes. And think about the metabolism of these compounds.
So it's because it's like a factor. Thiamine is a cofactor, right? Yeah. I didn't even detect positive right now. Look who's naturally good. That's not gonna happen. Yeah.
If you don't have cofactor binding that enzyme, it's not going to be special.
This is used to get pyruvate essentially all the way to right here. And so if you can't turn pyruvate those away, it's not just gonna sit there. And then the alpha-ketoglutarate make that shit build up.
There's no, I feel like there's like a build up. Because if the first cofactor's not working, that's not right.
I have like
Yeah, I do that first. I thought you like
Yeah, I tried. Where are the opposite? There's no opposite.
license.
Yeah. Oh, yeah, that's what happens.
But sometimes you explain stuff and it sticks in my head.
Well, what if it's wrong?
Okay. We'll find out next Monday. As long as it makes sense and I understand it.
So you can choose.
We can try. If we don't, if I don't understand it, it's fine.
Specific topics of physics. It breaks it down. But you don't take full 8 hours. And they want.
Oh, that's not funny.
Sorry. I got motion.
Okay, so you have to eat vitamin B1 to keep from getting very, very, and that's because thiamine is needed as a cofactor in a couple of enzymes. So what's one of those enzymes? Right, pyruvate dehydrogenase complex. The E1 component, the E1 subunit of pyruvate dehydrogenase complex, has a thiamine in its active site, thiamine pyrophosphate, and that's needed to accept the acetyl group in the decarboxylation of pyruvate. So without that, then that enzyme can't decarboxylate pyruvate. Pyruvate can't go into be converted to citric acid to acetyl-CoA to go into the citric acid cycle, so pyruvate accumulates in the blood. And because of that, then you can't use glucose properly because you can't metabolize it through the citric acid cycle. So there's another enzyme that I mentioned has very similar properties, works in the same way as pyruvate dehydrogenase. What's it? In the citric acid cycle. Alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase also has the E1, E2, E3 components and carries out a similar reaction as pyruvate dehydrogenase. It's a decarboxylation reaction and an oxidation. And that enzyme in its E1 component also has a thiamine pyrophosphate. So without that enzyme, alpha-ketoglutarate can't be metabolized, so it also accumulates in the blood. So this can be used as a blood test to determine that someone has beriberi. If they have high levels of pyruvate and alpha-ketoglutarate in their blood, it's because they're not able to metabolize pyruvate. Okay, I gave you a summary that I'm not going to read through that you can use to help you study, and I have a little video about the Krebs cycle.
The molecules of life had a mystery. He checks out the chemistry cleverly. When an air-breathing life form wants to make energy, the Krebs cycle's a main part of the remedy. Krebs, her rhymes get the name in your head. For his time spent and shuffled to the things that he did, and that's why he got to share the Nobel Prize for the citric acid cycle, the engine of life. When we eat, our body breaks the food into particles, providing the body with the daily dose of chemicals. We absorb the nutrients and utilize molecules to generate the energy that's needed in our body cells. We turn the food into ATP, a chemical that the cells use for energy. And there's a particular chain of events that we can see as part of cell respiration, why we need to breathe. The Krebs cycle is happening in all our cells. In the mitochondria, it's where the process dwells. It uses oxygen, so it's aerobic as well. It produces CO2 in every breath you exhale. It makes use of the products of glycolysis to get a 6-carbon molecule to put in the mix. And it's the citric acid molecule that prompted the title of Krebs discovery called the citric acid cycle. Krebs, the molecules of life had a mystery. He checks out the chemistry cleverly. When an air-breathing life form wants to make energy. Krebs cycles are made part of the gravity. Krebs, a rhyme to get the name in your head. For his time spent and shoveled in the things that he did, and that's why he got to share the Nobel Prize for the citric acid cycle, the engine of life. When Krebs was born back in 1900, details of metabolism hadn't been uncovered. He studied and then helped find the ornithine cycle. That's the first metabolic cycle to be discovered. His days were numbered as a Jew living in Germany. In 1933, he had to leave the place urgently. He spent a couple years down at Cambridge University, then moved over to Sheffield, where he lectured pharmacology. He got a new laboratory, they doubled up his salary, but he still had to focus on metabolism. Naturally, there had to be a process he could recognize, and in a few years, the pathways were identified. But right now, one of those is most relevant. It's called the Krebs cycle. It's the source of our energy, and it was an important discovery, getting him a Nobel Prize in physiology or medicine. Krebs, the molecules of life had a mystery. He checks out the chemistry cleverly. When an air-breathing life form wants to make energy, the Krebs cycles are made part of the remedy. Krebs, a rhyme to get the name in your head. For his time spent at Sheffield and the things that he did, and that's why he got to share the Nobel Prize for the citric acid cycle, the engine of life.
Okay, there's a link to that video in the PDF of the lecture if you want to watch it again with your friends. All right, so we're going to talk now about what happens to that NADH and FADH2 and ubiquinone that are generated in the citric acid cycle, and how they generate the energy for making ATP. So we're going to look at transport of electrons through a pathway that's going to result in oxidation of that NADH and FADH2 and recovering that energy to drive synthesis of ATP. So here's another question for you. Why do you need to breathe oxygen? And I want you to think of a biochemical explanation. What kind of reaction is that oxygen involved in? What is it converted to after you breathe it in? So talk to each other about that for a couple minutes. Okay, so the wrong answer is carbon dioxide. It's not converted to carbon dioxide. Where does carbon dioxide come from? Say, what pathway have we just talked about generates carbon dioxide? Citric acid cycle. So the carbon dioxide that you exhale comes from pyruvate dehydrogenase reaction and citric acid cycle. So it's oxidation of pyruvate and acetyl-CoA. There's no molecular oxygen in that reaction, that pathway. So it's not carbon dioxide. So what is the oxygen converted to? Water. That's right. So that oxygen is used as an electron acceptor. It's the final electron acceptor in the pathway we're going to talk about today, and it's the oxygen atoms are reduced to form water. So the pathway is called oxidative phosphorylation. It's oxidizing NADH and FADH2 and reducing at the end molecular oxygen to make water. And it's phosphorylation of an ADP to make ATP. And those are linked together through a pathway. So we talked about in glycolysis, we're starting with pyruvate, the product of glycolysis is pyruvate. And we're going through pyruvate dehydrogenase and citric acid cycle, oxidizing the carbons there to carbon dioxide and preserving those electrons, the reducing power in NADH and FADH2. So we're going to look at how oxidizing those cofactors is linked to synthesis of ATP. This takes place in the mitochondria. The proteins involved in the electron transport chain are in the inner mitochondrial membrane. Where does the citric acid cycle occur? Let me back up a step. Where does glycolysis occur? Cytosol. Okay. And where does pyruvate dehydrogenase? In the mitochondrial matrix, inside the mitochondria, and the citric acid cycle is in the same compartment. So pyruvate dehydrogenase and citric acid cycle are in the inside of mitochondria. So here's a cartoon of a mitochondria. It has two membranes. It has an outer membrane and an inner membrane. And the space inside the inner membrane is called the matrix. And so the citric acid cycle and pyruvate dehydrogenase enzymes are in the matrix. They're producing that NADH and FADH2 in the place that's in the same location as the proteins that are involved in the electron transport chain, which are in the inner membrane of the mitochondria. And there's a space between the inner membrane and the outer membrane that's going to become important as we talk about what those do, because those proteins are integral membrane proteins in the inner membrane, and they pump protons out into the space between the two membranes. So we'll be looking at that. Okay, so we're going to look at how electrons from NADH and FADH2 are transported through a series of steps where the final acceptor is oxygen. It doesn't directly transfer those electrons from NADH to oxygen. It goes through a number of steps, and those steps can be regulated, and they generate the energy that's used to pump the protons, that makes the stored energy that's used for ATP synthesis. And the proton gradient is achieved by pumping those protons from the matrix side out into the space between the membranes. Then when those protons flow back into the matrix, that's the energy that drives ATP synthesis. So we'll be looking at that. Okay, so we need to think a little more again about oxidation and reduction reactions. So remember that when a molecule donates electrons, when it loses electrons, it becomes oxidized. When it gains electrons, accepts electrons, it becomes reduced. So we're thinking about which molecule is gaining electrons, which one is losing electrons. We can measure that, the affinity of an electron acceptor for the electrons. And we can measure that and measure a standard for each kind of molecule, for each chemical. We can measure its standard reduction potential. What is its affinity for electrons? And so this is the unit for standard reduction potential. So it's like standard free energy change that you talked about. Did Dr. Zolk used to talk about standard reduction potential? Not remembering it. Okay. So it's related to standard free energy change, and I'll show you that in a minute. So this standard reduction potential has a unit in volts. So it's about a movement of electrons. It's measured in volts. And a molecule that has a strong, that is a strong reducing agent has a negative value for the standard reduction potential. So the molecules we're thinking about here to start, NADH is a strong reducing agent. So when we're writing out the equations for standard reducing potential, we write them in the direction of reduction, okay? So if NAD accepts 2 electrons to make NADH, that has a negative standard reduction potential. That means it's a good, NAD is a good reducing agent. NADH is a good reducing agent. Oxygen is a strong oxidizing agent. If we reduce oxygen to water, that has a positive value for standard reduction potential. Okay, so the net reactions we're thinking about is transferring electrons from NADH through a series of steps to oxygen to make water. And in that reaction, if we look at the net reaction, a half of an O2 plus an NADH to make a water and an NAD, we can calculate that standard reduction potential. This is the standard reduction potential for NAD to NADH, and we change the value of that because in this case, we're talking about oxidation of NADH instead of reduction. So it's a minus or a plus 0.32 volts. And the reduction potential for oxygen is 0.82 like we had here. So the total, if we sum together, those are 1.14 volts. So we can relate that to standard free energy change. Here's just the equations we've talked about so far to get the net 1.14 volts standard reduction potential for this reaction. The standard reduction potential is related to standard free energy change by this equation. Standard free energy change equal to minus N, which is the number of electron transfers. So that's going to be 2 in this reaction, times the Faraday constant, which is 96.5 kilojoules per volt per mole. You don't have to memorize that or this equation. And then the standard free energy change in this reaction, we calculated at 1.14. So if we take this and plug those numbers in, 2 electrons times the Faraday constant times the standard reduction potential, the standard free energy change for this oxidation of NADH and reduction of oxygen has a free energy change of minus 200 kilojoules per mole. So that's a big negative delta G. That provides a lot of energy for ATP synthesis. And so harnessing that energy is what we're talking about in this pathway. So we get minus 200 as standard free energy change for the oxidation of 1 NADH. For making an ATP from an ADP and a phosphate, that standard free energy change is about plus 30. So it requires an energy input to phosphorylate ADP to make ATP. And you can see then that by oxidizing NADH, we get plenty of free energy for making ATP molecules. And under the conditions in a cell, as I mentioned, that's enough energy for about 2.5 ATPs from each NADH. So oxidizing 1 NADH provides about enough free energy for 2.5 ATPs. The transfer of electrons isn't direct from NADH to oxygen. It happens through a series of steps that we call the electron transport chain. So electrons are transferred from NADH to one molecule, to another molecule, to another molecule, to another molecule, and the energy is harvested through those electron transport steps and preserved for ATP synthesis. So there's a chain of electron carriers in the inner mitochondrial membrane. We're gonna look at those in some detail. 4 of those are integral membrane protein complexes. So they're proteins embedded in the inner mitochondrial membrane. That means that they have to have the hydrophobic regions that pass through the hydrophobic regions of the interior of the lipid bilayer, and they span the bilayer, so there's some part domains of the proteins on the outside of the inner mitochondrial membrane and some on the inside. And they're protein complexes. They're composed of multiple protein subunits. One of those electron carriers is not a protein, it's a lipid. That's ubiquinone that we've already talked about. So ubiquinone is able to accept electrons from one integral membrane protein and transfer them to another. And one other electron carrier is not an integral membrane protein, it's a peripheral membrane protein. It's cytochrome C. So it's not embedded in the membrane, it's at the surface of the membrane, and it's able to move freely along the surface of the membrane from one protein to another to accept electrons and then to donate electrons to another protein. Okay, so this is a graph that shows the kind of the path of electrons in the electron transport chain and how the free energy changes as those electrons are transferred. So if we start with NADH, we transfer them to a protein complex called complex 1. It transfers them to ubiquinone that we've talked about. Ubiquinone transfers its electrons to complex 3. Complex 3 transfers electrons to cytochrome C. Cytochrome C transfers electrons to complex 4. And complex 4 is where oxygen is reduced to make water. So as we go along this electron transport chain, we're going to talk about these components one by one in a minute. We see a decrease in free energy of the electrons as those energy that was stored in those NADH and FADH2 are recovered in a proton gradient. Okay, so let's look at the first step. Electrons starting from NADH are transferred to complex 1. Complex 1 is an integral membrane protein complex, multiple protein subunits. And the longer name for complex 1 is NADH ubiquinone oxidose reductase. So it's accepting electrons from NADH, transferring them to ubiquinone. The complex 1 contains a number of prosthetic groups that participate in the electron transport. The first one of those is a flavin mononucleotide. So electrons are transferred from NADH first to this flavin mononucleotide to reduce it. Then the electrons are transferred from the flavin group to some sets of atoms in complex 1 that are called iron sulfur clusters. So there's a series of pathway of electrons transferred from one iron sulfur cluster to the next. Those iron sulfur clusters, the sulfur comes from cysteine in the protein. So remember that cysteine is an amino acid with a sulfur, a sulfhydryl group in its side chain. And those cysteines in the protein complex with an iron atom. And there are different sorts of iron sulfur complexes in complex 1. And we get then transfer, they're located in the protein complex at just the right distance for electrons to jump from one of these irons to another. And so the iron is being oxidized and reduced as the electrons are transferred from one iron to another, and the irons are held in place by cysteines that are part of the protein molecule. Okay, so this is my drawing of the electron transport chain that you're going to see a few times. We're right here now with complex 1. So NADH is in the matrix side. It's produced from pyruvate dehydrogenase and citric acid cycle. Complex 1 is going to oxidize that NADH back to NAD. and transfer the electrons to complex 1. Complex 1 then transfers electrons to ubiquinone. And you can see that then, we're going to talk next. Ubiquinone transfers its electrons to complex 3. We're going to look at what happens in between there. Okay. So in step 2, the iron sulfur clusters are going to donate those electrons to ubiquinone and reduce it to ubiquinol. And that happens also with electrons from FADH2. So electrons from FADH2 are produced in the citric acid cycle, and that's done by the enzyme, the succinate dehydrogenase. So remember, succinate dehydrogenase is an enzyme in the citric acid cycle that produces an FADH2. That actually, we call it complex 2 in the electron transport chain. So it's the same protein complex in citric acid cycle and in the electron transport chain. We call it complex 2, it's succinate dehydrogenase. So it transfers its electrons from FADH2 to ubiquinone to make ubiquinol. An important thing to see is that then the electrons from FADH2 skip the first protein complex. And so that's the reason that they're making less ATP. They provide less energy for making ATP because they don't go through complex 1, which is pumping protons to the space. Okay, so the electrons from FADH2 that come from succinate dehydrogenase, FADH2, are transferred to ubiquinone. Ubiquinone is a lipid. It can diffuse through the membrane, and it's going to move to complex 3 and donate its electrons to complex 3. So this is, again, the structure of the oxidized form of coenzyme Q, ubiquinone. It's a quinone with these double bonded oxygens in the ring. It accepts the electrons, and it accepts 2 electrons to make, then reducing both of those oxygens in the molecule to alcohols, and this is the ubiquinol form. The reduced form is what moves into the membrane and donates its electrons to complex 3 in the pathway. Okay, complex 3's name is ubiquinol cytochrome C oxidoreductase. So it's taking electrons from ubiquinol, donating them to cytochrome C, which is this small peripheral membrane protein. So electrons from complex 3 are donated to cytochrome C, which can move along the surface of the membrane, and then cytochrome C is going to be able to donate the electrons to complex 4. Complex 4 has another name, cytochrome C oxidase. So it's going to oxidize cytochrome C, and in the process, those electrons are transferred to oxygen to make water. Cytochrome C oxidase complex 4 has in it cytochromes, which are molecules that are related to heme, that are in hemoglobin. They have an iron that's involved in electron transport. And finally, also some copper ions. So we have electrons moving through a series of steps from those cytochromes to copper ions and finally to oxygen to make water. So this is a very important step in the electron transport chain. Step 4 oxidizes cytochrome C and reduces oxygen. There are 2 kind of famous poisons that inhibit complex 4, and that's cyanide and carbon monoxide. Both of those work as competitive inhibitors. They take the place of oxygen in the complex 4. So they occupy the space where oxygen would be and prevent this reduction of oxygen to water. And that's why they're such good poisons, is that they stop in the electron's transport chain, stop you from making ATP. Because without this step, you don't generate the energy for making ATP. So carbon monoxide is a byproduct of burning gas. So in my house, I have carbon monoxide detectors that if there's something malfunctioning in my furnace, that methane that's being burned in the furnace, if it's not getting enough oxygen, won't be completely oxidized and converted to carbon monoxide, which is you can't smell carbon monoxide, you can't tell it's there, and so that's why people who get poisoned with carbon monoxide, if there's generation of that in some kind of combustion reaction. And it's also the reason cyanide is such an effective poison, is that it poisons mitochondria. It stops mitochondria from reducing oxygen to make water. The gas company puts a molecule in the gas that, so the gas that's burned is mostly methane, but there's a molecule, mercaptoethanol, that's added to the gas in small amounts that has a sulfur smell. And so if your gas is leaking, you can smell that gas is leaking because of the mercaptoethanol. But if it's a carbon monoxide problem, you can't smell that. It doesn't have any odor. Okay, so the flow of electrons through the electron transport chain, that energy from the electron transport is recovered by complexes 1, 3, and 4. Complexes 1, 3, and 4 are proton pumps. They pump protons from the matrix side out into the space between the membranes and generates a proton gradient. So that proton gradient you can think of as something like energy stored, like water stored behind a dam. There's a high concentration of protons now in the inner membrane space and lower concentration of protons in the matrix. And the flow of those protons back into the matrix is what provides the energy for ATP synthesis. And that's done by a fifth membrane protein complex called ATP synthase, and we're complex 5. Okay, so here's the pathway. We're generating from citric acid cycle NADH and FADH2. The electrons from NADH are transferred to complex 1 and then to ubiquinone. That electron transfer causes pumping of protons through complex 1 out into the space between the two membranes. The electrons from FADH2 generated in the citric acid cycle are donated to coenzyme Q, which can move to complex 3. Complex 3 uses that electron transport to pump protons to the space. The electrons are transferred to cytochrome C, which then goes to complex 4, and complex 4 is also a proton pump. So three proton pumps in this pathway. And finally, electrons are donated to oxygen to make water. Any questions? Okay. I think we're going to stop there. We're going to go on to discuss how that energy is used to make ATP on Wednesday. I put a set of review items in the module on Canvas for you to look at. I think that you write out the answers to those in much detail as you